Hello, everyone, and welcome to our one-on-one discussions with the scientist. My name is Dalton Dietrich, and today I'm speaking with Dr. Pantelis Tsoulfas, who's an associate professor in the Department of Neurological Surgery and a very important component of the MY Project program. Hi, Pantelis. Thanks for joining us today.
Hi, Dalton. It's a pleasure to see you in the morning and looking forward to this interview.
Fantastic, fantastic. Well, obviously, Pantelis is very integrated into the project and doing a lot of exciting things, innovative things that we'll talk about today, including imaging and looking at different molecular mechanisms of regeneration. So Pantelis, one of the big problems we have in my project, or spinal cord injury in general, is trying to select what tracks after a spinal cord injury we should concentrate on to enhance regeneration. Those tracks are extremely complicated. Many times some tracks are very difficult to regenerate, where others may be a little bit easier, like the sensory tracks. So tell us exactly how you and your colleagues are going about trying to understand after spinal cord injury what tracks are injured and which ones really need our attention to enhance regeneration and repair.
Yeah, so thanks for the question, Dalton. So what has happened over the years, when you read papers, people tend to focus on specific tracks. The main one is the corticospinal tract that everybody's focused because of the importance to voluntary movement in humans. However, what happened is we have even more axons, more neurons that project down to the spinal cord, and these tracks are also important for movement. The question is why people look at one track. The issue is analysis. It takes a really long time to analyze a track, find all the actions that are spare, count it, and with traditional histological methods, takes weeks, months. So makes it impossible to look at all, if it can be done, at all the tracks that project to the spinal cord. So me and my colleague in Marquette, Murray Blackamore, we wanted to tackle this problem and not just look at one track, but look at all the neurons that project to the spinal cord. So that is the one of the main issues. The other thing is also we wanted to find techniques that can help us not only image the neurons, but also deliver molecules to all the neurons. So when we target a particular track, we just want to target all the tracks, not just one, corticospinal. So we can use it, so we can combine imaging and also therapeutic intervention. So we came up with some techniques, and we are in the process of actually writing the paper.
Fantastic. And those techniques include very innovative 3D reconstruction of the tracks.
Yeah.
Can you tell us exactly about that technology and what that's brought to the field?
Yeah, so I will backtrack first. First, we want to identify these neurons, and we publish a paper on Journal Neuroscience where we use some new viruses that got described a year before, and this is an adeno-associated virus that, when injected, is be taken by the terminals of these projections of these axons that go to the spinal cord, and then they transport back the virus to the body that is in the brain, and then they make a protein that we can look under the microscope. Mm-hmm. And we publish that. So the technological advance was the retrograde labeling first, and second is the imaging. And the imaging, this has been, how you can say, in the last 10 years, revolutionized the field of neuroscience, where you can label specific neurons and then clear the brain with specific chemicals, and then image in a microscope that is called a light sheet microscope, where allow us to look at a transparent brain, or at the whole transparent brain within 20 minutes. So we combine those two techniques, the retrograde labeling and the clearing techniques to do that.
And you can use different viruses. You can inject one virus before the injury and see exactly what the normal projection is, or going up to the supraspinal levels. And then you can actually injure the spinal cord injury, and then go back with a second virus and find out whether or not that particular track has been damaged or still actually transports the virus.
Exactly, Dalton. This is really a good comment, which was the next thing we're going to talk. And that is what we do. In the first phase of the study, we wanted also to count. It wasn't just looking qualitative where things go and they finish. We wanted to actually count how many neurons project at a specific level, and the neurons, let's say, from the corticospinal tract, from the hypothalamus, from the brainstem, and count all the major neurons. And so that is what we really did first. We injected at different levels, and then we went back and counted. So we have counts now, so we know how many corticospinal neurons project at each level. So we have a baseline, which is the first part of the thing you inject before the injury. So we know before the injury the approximate number, because obviously there is variability from animal to animal, so we will know the numbers. And next, which what we have done is do the injury at different severities, and then inject below the injury, and then look how many neurons depending on the injury level. So we have a baseline. So the next phase is actually use this approach to use interventions into the spinal cord to improve regeneration. For example, a stem cell transplant or some other chemical that I can influence the local environment, and see if the second virus that is injected after the intervention give us more neurons. So now we can count them. So we can count all the neurons. So we know, the corticospinal tract doesn't regenerate with this intervention, but the cerebrospinal regenerates, and we can count. So we can see what gets spared, what regenerates depending on the intervention. So we have a kind of global view.
Yeah, and a big advance has been with the 3D of being able to cut down the amount of time that it really takes to do these counts. Usually in the old days, we had to take semi-serial sections, and someone actually had to count each section.
Yes.
But now with the three-dimensional reconstruction, the computer can allow us to do more integrated and computer-assisted counting, which speeds up the science, which is all very good.
Yeah. So there is the time, and also a question of accuracy. The previous methods were really good, but they were an estimate, because you couldn't count anything. Now we can actually pinpoint, we can zoom in and say, oh, look, in this region, there is 3-4 neurons. And then, okay, this is the region where these particular nuclei, and so we can study that further. And I think we were lucky in part because you agreed to buy this microscope years ago. We bought this it's called ultra microscope, which allow us to image the whole brain. And I have to say, we were the first academic lab in the United States to purchase the instrument. So in that sense, we pioneered these methods, and the Miami project has published several papers using this technique, in which also at the end help us get grants because it's part of the innovation.
Yes. So yeah, that's one of the things the Miami project tries to do, is bring new tools and strategies for doing our science into the field. Another area that you're very interested in has to do with blood vessels and the vasculature of the spinal cord. Obviously, vasculature is really important throughout the nervous system in terms of delivering sources of energy and things of this nature, substrates. So we now know that after brain or spinal cord injury, there's a lot of injury to the blood vessels and then repair mechanisms. So tell us a little bit about the questions you're asking in that area of research.
Yeah, so the first approach we wanted to take is there were several papers over the past years where they identify genes that express in particular subsets of cells within the vascular unit. You have the lining, the endothelial cells, and then you have the mural cells, the cells that are around. So people have identified genes that are specific to each one of these cells. Further, they also found genes that can distinguish between the arterial system, which is the blood, the vessels that bring the oxygen to the spinal cord, and then the venous system that takes the blood that is not oxygenated anymore back to the lungs. So again, we use these techniques to image in 3D the spinal cord, the vessels, where are the arteries? A lot of description has been done over the decades, people know, but we wanted to look in 3D, big segments of the spinal cord, how the arteries come in, how they get distributed, and where is the venous system. So we are able actually to use some of these antibodies that recognize the specific populations. And we have some really beautiful images that I hope I can share with you quite soon. And in collaboration with Jay Lee, another investigator at the Miami project, we took this further a step, and he injured the spinal cords, and then at different time points went in and imaged in 3D how the vessels developed during injury from day 1 to day 21. And we have some data right now, and we're trying to quantify, it's really interesting what we see, which is suspected in these kinds of injuries as new vessels are formed and other vessels get destroyed. And so we have a nice view now. Now we need to quantify these phenomena. That is one aspect. And then the second aspect is, perhaps you already talked with Jay Lee on his work with the single cells. I have been involved intimately with this project, and my main interest has been the vessels.
Okay.
So we have some beautiful data on all these populations in the vessels, how they change over time, and what are the gene changes. So now we want to combine the 3D imaging from what we learned from the single cell and start manipulating. And because we can look in 3D, we can look after manipulation, what happens to the vessels, how they change. And so I think this is really a new way of looking at vessels, probing the gene changes at the single cell level and then imaging in 3D.
Yeah. And of course, the neurovascular unit is so important in terms of how the nervous system works. So understanding the neuronal changes versus the vascular changes after spinal cord is so important. So a lot of your work obviously is done in preclinical models, but ultimately you want to do some studies that translate into the human spinal cord. So we're very fortunate that my project has one of the largest human spinal cord collections in the world. And our vision is to utilize that particular material to actually do some of our cutting edge molecular research that we're doing. So tell us a little bit about how you're trying to move some of these techniques to human materials.
Yeah, so the idea again was to, there is a lot of new technologies, how we can expand our knowledge of spinal cord injury, because, the animal models are really valuable, but there might be differences. There is genetic heterogeneity between humans if each injury is different. And in the past studies, which some have been done at the Mayan project, and you guys published some with Dr. Nuremberg and others using these data, shown some of these changes. We wanted to take a step further and look again at gene changes. At this time, looking at particular pathways. And that is one aspect. The second is able to multiplex, practically look under the microscope, the gene changes or the protein changes, not just of 1 or 2 proteins, but 12, 30, 40 if we can. So we started a collaboration with a company that has pioneered some of these methods. It's called NanoString. We went and took some of these spinal cords and we extracted the RNA from the paraffin sections, and then we send it to them. The good news is that we get some data on a few of the spinal cords, but in others we couldn't, I think because maybe the material is not the best for RNA because it's collected after a day. And so we thought now maybe we can really actually switch from the RNA because of the technical difficulties, to the multiplexing, looking at 12 proteins or 30 proteins all at once. And we started this project, and we were working on it before the COVID-19 hit us. So we have put them a little bit on the back burner now because we can't go into the lab and do the experiments. But so the idea is to image these spinal cords by using proteins that recognize the astrocytes, the inflammatory cells, the neurons, the glia cells, and then we can look in one section at changes for 12 up to 35 proteins. This way we can get a better idea of what is going on. So that is what the project is about.
Yeah, I think that's so important. We continue to collect samples, so hopefully we'll have samples that are better materials so that you can actually do these studies more efficiently. So anyway, you mentioned the COVID-19 pandemic, and we're in the middle of it right now. So how are you continuing some of these studies and when it's not easy to go into work and it's not easy to actually do bench work?
So I actually go into the lab. When they're asking for letters for essential experiments, I explain that continuing to clear the brains, because I have a lot of brains I have to clear. And so actually I go to work sometimes 4 days, sometimes 5 days. I go into the lab and just do the experiments. There are big intervals in the meantime. I try to work on the computer and so on. And then 2 days I spend on the microscope imaging, collecting the data, and then send it to the collaborators. Actually, I do go to work. But I also, try to do as much as I can on the computer.
Yeah. Social distancing, all the things that we're doing. We're trying to be safe as we start slowly getting back into the laboratory. I certainly appreciate all the literature that you send me. I know you do a lot of searches and look at the general literature in terms of a lot of different topics, which I really appreciate. Pantelis, thank you very much for spending some time with us today and talking about some of your research interests that are very important, and wish you all the best for the future. And again, have a great day. Thank you very much.
Thank you, Dalton. Thank you. Thank you for spending time with me. Thanks.